imagine the world with your eyes closed to very small things. For centuries, the idea that unseen creatures might cause decay, change, and illness was debated, half claimed, and half feared. The story of microbiology begins when the invisible finally becomes visible. Welcome to the Sleepless Academic. Tonight we'll take a gentle, guided walk through the hidden world of microbiology, the tiny life that shapes our bodies, our planet, and the future of medicine. If you're listening as you settle into bed, tell me in the comments where you're listening from and whether it's morning, evening, or somewhere quietly in between. If you enjoy drifting off while learning, subscribe and join our community of curious sleepers. The Birth of Microbiology, Seeing the Invisible. Microbiology is, at its core, the study of the unseen. The term comes from the Greek words micros, meaning small, bios, meaning life, and logos, meaning study. Together they form the study of microscopic life. This field encompasses an astonishing variety of organisms: bacteria, archaea, fungi, protozoa, algae, and viruses. Each group is distinct, yet interconnected, shaping ecosystems, human health, and the planet's chemistry in profound ways. To understand microbiology is to realize how invisible processes dictate visible outcomes. For instance, the bread rising in your kitchen owes its texture to yeast, a microorganism converting sugar into carbon dioxide bubbles. The nitrogen in your body, vital for DNA and proteins, once passed through bacterial hands, transformed from atmospheric nitrogen into a usable form. Even the oxygen you breathe, nearly half of it, emerges from cyanobacteria drifting in oceans. Historically, humanity's awareness of this hidden world is very recent. For most of history, people attributed disease to imbalances of the four humours, to bad air or miasma, and sometimes even to spiritual punishment. It wasn't until the invention of the microscope in the 17th century that scientists caught their first glimpse of microbes. In the 17th century, a tradesman and lens maker named Antony van Leeuwenhoek polished simple lenses and used them to peer into drops of pond water, scrapings from his teeth, and other tiny worlds. He described animalcules, living shapes writhing and swimming, and sent his observations in letters that would eventually change how humans thought about life. From then on, the possibility that there existed entire unseen ecosystems around us became harder to deny. A second revolution came with careful experiments. Louis Pasteur in 19th century France showed that life did not spontaneously arise from broth, left to sit in a sealed, sterile container. He demonstrated that microbes come from other microbes, overturning spontaneous generation thinking, and laying the groundwork for sterilization, pasteurization, and the concept that microbes cause fermentation and disease. Robert Koch, a contemporary, built on these ideas and devised rigorous ways to link a specific microbe with a specific disease, rules that guided clinical microbiology for a century. These historical moments, observation, experiment, and proof, form the backbone of the field we now call microbiology. From those origins, microbiology split and spread. It is both a basic science, asking what microbes are and how they live, and an applied science, learning how to control them for health, food, and industry. Microorganisms include bacteria and archaea, single-celled prokaryotes, fungi and protozoa, eukaryotic microbes, microscopic algae, and viruses, which are simpler and require host cells to replicate. Though diverse in form and function, these tiny organisms share one defining attribute. They operate at scales too small for unaided sight, yet their combined actions sculpt ecosystems, soils, and human bodies. The fundamental difference. Prokaryotes and Eukaryotes. At college level, you'll often begin by separating life into fundamental categories. Prokaryotes, bacteria and archaea, are usually single cells without a nucleus. Their genetic material is not enclosed within a membrane-bound nucleus. Eukaryotes, fungi, protozoa, plants, animals, have nuclei and a richer internal architecture, organelles like mitochondria, and, in photosynthetic eukaryotes, chloroplasts. This structural distinction influences everything from reproduction to metabolism to how we target microbes with drugs. Seeing microbes, microscopy and staining. A microbiology lab course introduces microscopes early. Light microscopes reveal cell shapes and arrangements. Phase contrast and dark field methods help visualize living cells without heavy staining. Electron microscopy allows us to see surfaces and internal details at the nanometer scale. Viral shapes, membrane structures, and subcellular complexes. Staining techniques, notably gram staining, group bacteria into gram-positive or gram-negative depending on cell wall structure. This simple binary can predict behavior, influence which antibiotics may work, and orient clinical decisions. Culture techniques, growing microbes on media. Let us isolate species, study their behavior, and test drug sensitivity. Modern labs complement these classical techniques with molecular tools like PCR and DNA sequencing, which let us identify microbes by their genetic signatures even when they won't grow on plates. Laboratory practice and safety, the unseen rules. A college microbiology class doesn't only teach theory, it trains careful hands, sterile technique to avoid contamination, biosafety rules to protect students and prevent environmental release, and ethical practices for handling clinical samples. A small procedural habit, passing a flame along a glass loop, labelling plates precisely, or keeping a tidy bench, translates into reliable data and safety. These practices are foundational. The microscopes and genomes are only as useful as the discipline behind them. The many forms of microbes. The microbial world is astonishingly diverse. Though invisible, these organisms outnumber and outweigh all other forms of life combined. Let's slowly journey through their major groups, pausing at each one as though we are visiting exhibits in a vast, quiet museum of life. Bacteria. Bacteria are single-celled organisms with remarkable versatility. They lack a nucleus, which classifies them as prokaryotes. Their shapes are often simple: spheres, cocci, rods, bacilli, and spirals, spirilla. Yet these forms belie their complexity. Some bacteria thrive in extreme heat, others in acidic springs, and still others inside your digestive tract. Beneficial bacteria, like lactobacillus, help digest food and produce vitamins. Others, like Escherichia coli, are normally harmless but can sometimes become pathogenic. Archaea. Closely resembling bacteria in appearance, archaea are genetically distinct. They often live in extreme environments, boiling geysers, hypersaline lakes, or even deep-sea hydrothermal vents. Their resilience makes them fascinating models for the study of life's limits. Some archaea produce methane, a process used in waste treatment and energy production. Others suggest what extraterrestrial life might look like, surviving where few others can. Fungi. Fungi are eukaryotic, meaning their cells contain nuclei. They range from single-celled yeasts to complex molds and mushrooms. In ecosystems, fungi are decomposers, recycling dead material into nutrients. Yeasts ferment sugars into alcohol and carbon dioxide, central to bread, beer, and wine. Penicillium molds gave rise to penicillin, the first widely used antibiotic. Yet some fungi also cause disease, from athlete's foot to systemic infections. Protozoa. Protozoa are single-celled eukaryotes, often motile. They move with cilia, flagella, or pseudopodia. Many live freely in water, but some are parasitic. Plasmodium, for instance, is the protozoan responsible for malaria, a disease that continues to shape human history. Algae. Microscopic algae may seem like plants, but they are distinct. They perform photosynthesis, producing oxygen, and forming the base of aquatic food webs. Cyanobacteria, though technically bacteria, play a similar role, helping to oxygenate Earth's early atmosphere billions of years ago. Viruses. Viruses challenge our definition of life. They are not cells, but packets of genetic material, DNA or RNA, wrapped in protein. They cannot reproduce on their own, but hijack the machinery of host cells. Some cause mild infections like the common cold, while others unleash global pandemics. Viruses also play roles in evolution, inserting their genetic material into host genomes. A significant portion of human DNA comes from ancient viral remnants, prions and other infectious agents. Even stranger are prions, misfolded proteins that cause diseases like mad cow disease. Unlike viruses, they contain no genetic material, only a corrupted shape that spreads like a whisper through the body's proteins. Microbial genetics and molecular biology. How the small world stores and shares information. If Chapter 1 described how we first saw microbes and learned their basic shapes, Chapter 2 opens the instruction manual, the genetic and molecular machinery that makes microbial life possible. Microbes are compact, efficient genetic systems. They mutate, share, and remix genetic material in ways that let them adapt quickly, a feature that can be both wondrous and troubling. DNA, RNA, and the central dogma in microbes. At the foundation of life, whether in humans, plants, or microbes, lies nucleic acid, the molecular blueprint that encodes all the instructions for sustaining, reproducing, and adapting life. In most microbes, DNA serves as this central instruction manual, a long, elegant polymer storing information in sequences of 4 nucleotides: adenine, thymine, cytosine, and guanine. Some viruses, however, are exceptions to this rule. They carry RNA instead of DNA as their genetic material, a choice that allows them to replicate quickly and sometimes evade cellular defenses more effectively. The principle governing the flow of genetic information is known as the central dogma of molecular biology. In its simplest form, it states that DNA is transcribed into RNA, and RNA is then translated into protein. Proteins, in turn, perform the vast majority of cellular functions. They build structures, catalyze reactions, and regulate processes that keep the microbial cell alive and responsive. This central dogma is remarkably conserved across life, a testament to its efficiency and universality. Yet microbes add their own fascinating variations to this basic framework. In bacteria, for instance, groups of related genes are often organized into operons, clusters controlled by a single promoter. This means that multiple proteins required for a common function, such as breaking down a particular sugar or responding to a stress, can be switched on or off together. It's an elegant, economical system. Instead of regulating each gene individually, the microbe can respond as a unit, saving energy and ensuring that coordinated processes happen simultaneously. This efficiency is crucial because many microbes inhabit environments that change rapidly. Nutrient availability, temperature, pH, and the presence of competing organisms can fluctuate in minutes or hours. A bacterium that can quickly turn on the genes for metabolizing a new sugar, or switch off a pathway that is no longer needed, gains a survival advantage. Microbes have evolved sophisticated regulatory mechanisms, including repressors, activators, and small regulatory RNAs, to fine-tune gene expression with exquisite sensitivity. Viruses, by contrast, often adopt a more minimalist strategy. Because they rely entirely on host machinery for replication, their genetic material is usually compact, encoding only the essential proteins needed to invade cells and replicate. Some RNA viruses even carry enzymes like reverse transcriptase, which allows them to transcribe RNA back into DNA, effectively bypassing parts of the classical central dogma. This unusual strategy is famously used by retroviruses like HIV, giving them unique evolutionary flexibility. Beyond transcription and translation, microbial genetics also includes mechanisms for gene transfer. Bacteria can acquire new genes from their neighbours through horizontal gene transfer, a process that accelerates adaptation. Conjugation involves direct transfer via a pilus. Transformation allows uptake of free DNA from the environment, and transduction uses viruses as carriers. These mechanisms make microbial evolution highly dynamic, enabling the rapid spread of traits such as antibiotic resistance across populations. In studying microbial DNA and RNA, scientists also uncover layers of regulation that go far beyond the central dogma. Epigenetic modifications, RNA interference, and feedback loops allow microbes to respond with precision to environmental cues. This adaptability is why a seemingly simple bacterium can survive in a desert, a hot spring, or inside a human gut, all by reading, interpreting, and acting on the information encoded in its genetic material. So, when you imagine the microscopic world, picture a constantly active library of DNA and RNA, where each microbe reads its instructions, decides which genes to express, and adapts in real time to its surroundings. The central dogma provides the framework, but the ingenuity of microbial regulation ensures that life can persist, thrive, and evolve under conditions that would challenge even the most complex multicellular organism. Plasmids and mobile genetic elements. Bacterial genomes are often accompanied by plasmids, small circular DNA molecules that replicate independently. Plasmids commonly carry genes that confer useful traits, antibiotic resistance, heavy metal tolerance, or novel metabolic pathways. Plasmids can transfer between cells, spreading traits across populations. Beyond plasmids, bacteria exchange DNA via mobile elements such as transposons and integrative conjugative elements. Collectively, these facilitate rapid evolution by shuffling useful genes where they're needed. Horizontal gene transfer, so borrowing instead of waiting. One of the most significant differences between microbial evolution and our classic image of linear descent is horizontal gene transfer, HGT, the movement of genetic material across lineages. HGT occurs by three primary mechanisms: transformation, the uptake of naked DNA from the environment; transduction, the DNA transfer mediated by bacteriophages, viruses that infect bacteria; conjugation, the direct cell-to-cell transfer of DNA through a physical connection, often mediated by plasmids. Because of HGT, a beneficial gene can spread through a population very quickly, sometimes across species boundaries. This is a major driver of the rapid rise of traits like antibiotic resistance. Mutations, selection, and the evolution of resistance. Mutations, which are random changes to DNA, arise during replication or from environmental damage. In large microbial populations, rare beneficial mutations will appear frequently enough that natural selection can act rapidly. When selection pressure exists, for example, antibiotic exposure, genetic variants that confer survival advantages proliferate. This combination of high population numbers, rapid reproduction, and gene-sharing mechanisms is why microbes adapt so fast. The public health consequence, the rise of drug-resistant bacteria, is a pressing global concern. estimates and surveillance data show antimicrobial resistance already causes significant mortality worldwide and is recognised as a top public health threat. CRISPR, microbial memory turned genetic toolkit. One of the most elegant discoveries in recent microbial genetics is the CRISPR-Cas system. Initially discovered as an adaptive immune defense in bacteria and archaea, CRISPR systems capture short fragments of invading viral DNA and store them in the host genome as spacers. If the same invader returns, the stored sequences help the cell recognize and cut the invader's DNA. Scientists recognize this system could be repurposed as a precise genetic editing tool in larger organisms. By programming the CRISPR machinery to target specific sequences, we can cut and edit DNA with unprecedented ease. In contemporary university courses, CRISPR appears both as a fascinating example of microbial defense and as a cornerstone of modern molecular biology and biotechnology. Modern molecular tools, PCR, sequencing, metagenomics. Traditional culture methods reveal only a fraction of microbial diversity. Modern molecular approaches have rewritten our view. Polymerase chain reaction, PCR, amplifies tiny amounts of DNA so they can be analysed. DNA sequencing, once slow and expensive, is now rapid and cheap. Whole genomes and environmental DNA samples are routine data sources. Metagenomics, sequencing DNA from entire communities without culturing, reveals microbial dark matter, the vast majority of microbial species that we cannot yet grow in the lab. These tools allow us to survey microbiomes, the communities of microbes living in soils, oceans and inside bodies, trace outbreaks by genome fingerprinting, and discover new enzymes and metabolic pathways. From bench to bedside, genetic methods in diagnostics and therapy. In clinical microbiology, rapid detection matters. Molecular tests detect pathogen DNA, or RNA, faster than culture. Sequencing tracks outbreaks, identifies transmission chains, and helps design targeted therapies. Therapeutically, CRISPR-based techniques open possibilities, editing bacteria, engineering phages, or even modifying human cells, but these are still active areas of research with important safety and ethical questions. Microbial physiology and metabolism. How microbes eat, grow, and shape worlds. While genetics explains the instruction set, physiology explains how microbes live, their metabolic strategies, energy flows, and community behaviours. Nutritional categories and metabolic strategies. Microbes are astonishingly versatile. We categorise them by how they obtain energy, and carbon phototrophs capture light energy, photosynthetic bacteria and algae. Chemotrophs harvest chemical energy, oxidizing organic or inorganic molecules. Autotrophs fix carbon from CO2, like some cyanobacteria and chemoautotrophs. Heterotrophs consume organic carbon, many bacteria and fungi. Within chemotrophs, we further subdivide into organotrophs, using organic electron donors, and lithotrophs, using inorganic donors like hydrogen, sulfur, or iron. These metabolic categories explain why microbes occupy nearly every environment on Earth, from sunlit ponds to deep-sea vents where chemosynthetic bacteria fix carbon using energy from Earth's interior. Respiration and fermentation. Cells extract energy by moving electrons through chains. In aerobic respiration, electrons flow to oxygen via an electron transport chain, driving ATP synthesis via membrane-bound complexes. Anaerobic respiration uses different terminal electron acceptors, nitrate, sulfate, even metal ions. Fermentation is a simpler pathway used when electron acceptors are limited. Organic molecules serve as both donors and acceptors, yielding less energy, but allowing survival in oxygen-poor environments. Many microbes switch between modes depending on availability, a metabolic flexibility that helps them survive fluctuating conditions. Biofilms and communal life. Microbes rarely live alone. On surfaces, teeth, catheters, rocks, or pipeline walls, they form biofilms, structured, surface-attached communities embedded in self-produced extracellular matrices. Biofilms protect resident microbes from environmental stresses and antibiotics, facilitate nutrient sharing, and create microenvironments, oxygen gradients, pH niches, that support diverse species. Clinically, biofilms are important because they underlie chronic infections and are more resistant to treatment than free-living cells. Quorum sensing, the language of microbes. Many bacteria communicate chemically, a process called quorum sensing. Cells produce and release small signal molecules. When concentrations reach thresholds reflecting high cell density, coordinated changes in gene expression occur: biofilm formation, virulence factor production, bioluminescence. This collective behavior is an elegant example of how single-celled organisms coordinate at the population level. Extremophiles and environmental microbiology. Microorganisms inhabit extremes: high heat, thermophils near hydrothermal vents, extreme acidity, intense salinity, and high radiation. These extremophils teach us about life's limits, inform biotechnology, robust enzymes for industry, and even guide astrobiology. If microbes can thrive in Earth's extremes, perhaps life might exist in similar environments elsewhere. Applied metabolism, fermentation, biotechnology, and beyond. Humans have long harnessed microbial metabolism. Fermentation produces bread, beer, cheese, and yogurt. Modern applied microbiology goes further. Microbes synthesize insulin, produce biofuels, and help clean up pollution through bioremediation. Synthetic biology engineers metabolic pathways into microbial chassis to manufacture drugs, chemicals, and materials. Understanding metabolism, the flow of electrons and atoms through cellular networks, is the key to these applications. Medical microbiology and immunology. Medical microbiology is perhaps the most dramatic branch of microbiology because it places us face to face with the reality of infection, illness, and the microscopic battles happening inside our bodies every day. While microbes are essential to life, some have evolved strategies that allow them to invade human hosts, cause damage, and sometimes overwhelm our defences. At the same time, our immune system has evolved equally sophisticated defences to protect us. In this chapter, we will explore the causes of disease, the interactions between microbes and hosts, the remarkable complexity of the immune system, and the tools we use, vaccines and antimicrobials, to tip the balance in our favour. Pathogenic bacteria and viruses, how they cause disease. A pathogen is any microorganism capable of causing disease. Not all microbes are harmful, but those that are pathogenic have special traits that allow them to establish themselves in a host. Bacteria can cause harm in several ways. Some invade tissues directly, multiplying inside organs where they do not belong. Others release toxins, powerful chemicals that damage cells or disrupt biological processes. For example, Clostridium tetani, the bacterium responsible for tetanus, produces a neurotoxin that interferes with muscle relaxation, leading to painful spasms. Vibrio cholerae, the cause of cholera, secretes a toxin that causes cells in the intestines to pump out water, leading to life-threatening dehydration. Viruses operate differently. They are not complete living cells, but instead genetic material encased in a protein shell. To replicate, they must infect a host cell and hijack its machinery. The cell, forced to produce viral components, often bursts apart, spreading more viruses into surrounding tissues. This process is responsible for the symptoms of many viral illnesses, from the common cold to more severe infections like influenza. Some viruses, like herpes simplex, can hide quietly in cells for years before reactivating, while others, such as HIV, integrate themselves into the host genome and fundamentally alter immune function. Host-pathogen interactions, virulence factors and immune evasion. The relationship between a pathogen and its host is a dynamic battle. Pathogens do not simply invade blindly. They come armed with virulence factors, traits that enhance their ability to cause disease. Some bacteria are shielded by a capsule, a slimy outer covering that makes it difficult for immune cells to engulf and destroy them. Others secrete enzymes that break down tissues, creating pathways to spread deeper into the body. Certain pathogens can change the proteins on their surfaces, disguising themselves so the immune system cannot recognize them. Viruses also employ tricks of survival. Influenza virus, for example, mutates rapidly, producing slightly different surface proteins each season, which is why flu vaccines must be updated regularly. HIV is even more cunning, targeting the very immune cells, T helper cells, that should be organizing the defense, leaving the body vulnerable to other infections. This constant back and forth between host defenses and microbial strategies is sometimes called an evolutionary arms race. Each side adapts, counter-adapts, and develops new methods of survival. The immune system, innate and adaptive defenses. To resist pathogens, humans rely on the immune system, which operates at two interconnected levels, innate and adaptive immunity. The innate immune system is our first line of defense. It includes physical barriers like the skin and mucous membranes, chemical barriers such as stomach acid and antimicrobial enzymes in saliva, and cells like macrophages and neutrophils that attack invaders broadly, without needing prior exposure. These defenses are fast, general, and always present. The adaptive immune system is slower to activate but far more specialized. It learns to recognize specific features of pathogens, called antigens, and then mounts a precise response. B cells produce antibodies, proteins that bind to pathogens and neutralize them or mark them for destruction. T cells, on the other hand, have multiple roles. Helper T cells coordinate immune activity, while cytotoxic T cells directly destroy infected cells. What makes the adaptive system so powerful is memory. Once it encounters a pathogen, it remembers. If the same microbe appears again, the response is faster and stronger. This is why many childhood illnesses like chickenpox usually occur only once. The immune system, once trained, rarely forgets. Vaccines, teaching immunity in advance. Vaccines take advantage of this memory system by exposing the body to a harmless version of a pathogen, such as a weakened virus, an inactivated bacterium, or even just a fragment of protein. The immune system can prepare for future encounters. It's like showing the body a wanted poster of a criminal before the attack happens. When the real pathogen appears, the immune system recognizes it immediately and launches a rapid defense, often preventing illness altogether. Vaccines have eliminated smallpox, drastically reduced polio, and continue to prevent countless deaths from diseases like measles, whooping cough, and influenza. Newer technologies such as mRNA vaccines represent a breakthrough. Instead of delivering part of a virus, they deliver instructions for our cells to make a small piece of the viral protein, training the immune system without using any live pathogen. The success of mRNA vaccines during the COVID-19 pandemic demonstrated how flexible and powerful this approach can be. Antimicrobial therapies, weapons against infection. While vaccines are preventive, antimicrobial therapies are used to treat infections once they occur. Antibiotics, discovered in the 20th century, revolutionized medicine. Drugs like penicillin kill bacteria by interfering with cell wall synthesis, while others disrupt protein production or DNA replication. Antifungal drugs target features unique to fungal cells, and antivirals interfere with stages of viral replication. However, microbes are adaptable. The widespread and sometimes careless use of antibiotics has led to resistance, where bacteria evolve mechanisms to survive drug treatment. Some produce enzymes that break down antibiotics. Others pump drugs out of their cells or alter their internal machinery to make drugs ineffective. This has given rise to so-called superbugs, bacteria resistant to multiple classes of antibiotics. In hospitals, these infections can be deadly, as standard treatments fail. Addressing antibiotic resistance requires both scientific innovation, developing new drugs and therapies, and careful stewardship, ensuring existing treatments are used wisely. The delicate balance. Medical microbiology and immunology reveal a delicate balance between harm and protection. On one side are microbes equipped with tools to invade and exploit. On the other side stands the human immune system, endlessly adaptable, supported by medical interventions like vaccines and antimicrobials. The struggle is not one we can ever fully win, because pathogens evolve as quickly as we discover new defences. But through understanding these interactions, we tilt the balance in our favour, saving lives and pushing back the frontiers of disease. In this ongoing battle, we're reminded that our health is not guaranteed but constantly negotiated in the microscopic realm. The knowledge we gain in medical microbiology equips us not only to fight illness, but also to appreciate the resilience of both microbes and humans. Environmental and applied microbiology. While medicine often frames microbes as enemies, the environment reveals them as indispensable allies. Environmental and applied microbiology study the countless ways microbes support ecosystems, industry, and even our own bodies. Microbes in ecology, recycling the planet. Microorganisms are central to Earth's nutrient cycles. Nitrogen-fixing bacteria, like Rhizobium, live in nodules on plant roots, transforming inert nitrogen gas into ammonia, a form plants can use. Without this process, life would starve of essential proteins and DNA. In oceans, phytoplankton, microscopic algae and cyanobacteria drift in sunlit waters, producing half the world's oxygen and forming the foundation of marine food chains. In soils, decomposers like fungi break down organic matter, returning carbon, sulfur, and phosphorus back to circulation. Even greenhouse gases are influenced by microbes. Some consume methane, others release it, tipping the scales of climate balance. Industrial applications, harnessing microbial power. Humanity has long exploited microbes for industry. Fermentation, one of the oldest processes, relies on yeast converting sugars into alcohol and carbon dioxide. Bread rises, beer bubbles, and wine ferments thanks to yeast at work. Bacterial fermentation creates yogurt, cheese, sauerkraut, and soy sauce. Modern industries go further. Microbes produce antibiotics like penicillin, life-saving drugs that transformed medicine. Genetically engineered bacteria manufacture human insulin for diabetes treatment. In environmental cleanup, specialized microbes digest oil spills or detoxify heavy metals, a process called bioremediation. Microbiome studies, gut microbes and human health. One of the most transformative areas in recent decades is the study of the human microbiome, the trillions of bacteria, fungi, and viruses living in and on us. The gut microbiome in particular plays essential roles, digesting fibre, producing vitamins, and interacting with the immune system. Disruptions in this delicate ecosystem link to conditions ranging from obesity to depression. Research suggests a gut-brain axis, where microbial signals influence mood, cognition, and behaviour. Probiotics and prebiotics attempt to restore or support microbial balance. highlighting that we are not individuals but ecosystems of many species coexisting. Synthetic biology, engineering life. Perhaps the most futuristic application is synthetic biology, deliberately designing microbes with new functions. Scientists have created bacteria that glow in the dark, yeast that produce antimalarial drugs, and microbes engineered to capture carbon dioxide. In agriculture, engineered microbes improve soil fertility or protect crops from pests. Synthetic biology is both promising and controversial. It holds potential for medicine, energy, and sustainability, but raises questions about safety, ethics, and ecological impact. Still, it underscores the idea that microbes are not just natural forces but tools we can shape to meet human needs. Environmental and applied microbiology remind us that microbes are not only threats but partners. They sustain ecosystems, power industries, and even help define our health. They are in many ways the invisible infrastructure of life on Earth. Current and emerging topics. Microbiology is not a static field. It evolves alongside global challenges, emerging diseases, and new technologies. Current topics studied in universities today reflect this urgency and discovery. The antibiotic resistance crisis. Superbugs. Antibiotic resistance is among the most pressing threats to global health. Overuse of antibiotics in medicine and agriculture has fueled the rise of resistant bacteria. Methicillin-resistant Staphylococcus aureus, MRSA, spreads in hospitals. Multi-drug-resistant tuberculosis endures treatment regimens. Some strains of Klebsiella pneumonia resist even last-resort drugs like carbapenems. This crisis forces scientists to rethink strategies, developing new drugs, reducing unnecessary antibiotic use, and exploring alternatives such as bacteriophage therapy. Without intervention, projections suggest resistant infections could kill millions each year by 2050. Viral pandemics, influenza, HIV, and COVID-19. Viruses remain constant global challenges. Influenza mutates so quickly that vaccines must be updated annually. HIV, discovered in the 1980s, continues to affect millions, though antiretroviral therapy has transformed it from fatal to manageable. Most recently, the COVID-19 pandemic revealed how a novel coronavirus could reshape societies overnight. It highlighted the power of genomic sequencing, global cooperation, and mRNA vaccine technology, which may transform how we respond to future outbreaks. Phage therapy, old ideas and new hope. Bacteriophages, viruses that infect bacteria, are emerging as alternatives to antibiotics. Phages can target specific bacterial pathogens, sparing beneficial microbes, unlike broad-spectrum antibiotics. While discovered in the early 20th century, phage therapy was overshadowed by antibiotics. Today, as resistance grows, interest is resurging. Experimental treatments have saved patients from otherwise untreatable infections. Microbial dark matter, the unseen 99%. Most microbes cannot be cultured in the laboratory with traditional methods. These uncultured organisms, sometimes called microbial dark matter, represent the vast unknown. Advances in metagenomics, sequencing DNA directly from environments, now allow scientists to explore these hidden worlds. Each discovery reveals new branches of life, expanding the microbial tree and hinting at functions we barely understand. Space microbiology. Life beyond Earth? Microbes are also at the frontier of space science. On the International Space Station, microbes adapt in surprising ways, sometimes becoming more resistant to stress. Studying them helps protect astronauts' health and test theories about life in extreme environments. Some scientists explore whether microbes could survive interplanetary travel, perhaps carried on asteroids, an idea known as panspermia. Others consider engineered microbes as essential partners for future colonies on Mars, recycling waste, producing oxygen, or generating fuel. Medical, environmental, and cutting-edge topics together reveal microbiology as a field at the crossroads of health, ecology, and the future. It is a discipline not only about what is small, but about what is powerful, shaping human survival and the destiny of our planet.